The session today is going to be on neuron development and regeneration, and we'll be starting with Julia Kaiser at the Burke Medical Research Institute. She'll be talking about development of cortical projections to the brainstem versus cervical spinal cord. All right, go for it, Julia.
Yeah, let me share my screen. Someone else is sharing. Is it okay if I just stop? I guess so. Where do we have the screen? Is it this one? Maybe? Can you see the full PowerPoint, or is this just the?
There we go.
Okay, great. Okay, so as I was introduced already, hi, my name is Julia Kaiser. I work at the Burke Neurological Institute, and I'm going to introduce you a little bit into the development of cortical projections to the brainstem versus the spinal cord. First of all, I need to introduce you to this little guy, the corticospinal tract. It's the major descending tract of the cortex that controls our motor skills, and I think this is a function that we all highly appreciated during the quarantine. And in brief, upper motor neurons that are in the cortex project down to the spinal levels, where they directly or indirectly innervate lower motor neurons that then control the muscles that allow us to hold glasses and enjoy life. And as you can imagine, a lesion to this corticospinal tract by any of the mentioned or many other diseases or neurological conditions will leave the patient in, have a devastating impact on the quality of life and leave the patients at a point where they need care and assistance throughout their life. So that's obviously something that we want to be able to help them with in the future by regenerating. One thing that is also known about the corticospinal tract, as for many other cortical areas, is that there is a so-called somatotopy to it, meaning that certain areas in the cortex and also in the corticospinal tract specifically control different parts of the body. And it does that by actually extending the axons to different sites and target sites within the body, for example, found in the brainstem or in the spinal cord on cervical and lumbar levels. So far, it is not clear yet how this segmental axon targeting is specified during development, or in other words, how does a corticospinal neuron know what it wants to be when it kind of grows up? So that is a question that we are addressing in the Sahni lab. And my supervisor, Vibhu Sahni, showed in his previous research that there are actually at least two subpopulations of the corticospinal tract that extend their axons either to the cervical spinal cord or to the thoraco-lumbar spinal sections. And that there's also a transition zone between the cervical and the thoracic spinal cord where axons make a molecularly driven decision to either stop in the cervical spinal cord or to cross this transition zone into the lower levels, like the thoracic and lumbar spinal cord. He established that this decision is made early on in development, but it is maintained into adulthood, suggesting that there's a dedicated molecular program that steers this. And indeed, he found that thoraco-lumbar projecting corticospinal neurons can be identified by the factor Crim1, and that a cervical neuron, if it wants to not cross over this transition zone, needs to repress Crim1. So far, so good. He looked at this mainly based on the fact that the cervical spinal cord neurons are known to innervate hand functions or control hand functions, while lumbar spinal cord projecting neurons are more responsible for leg functions. And similar to that, we actually have another hotspot of motor control, namely the brainstem, where we know that facial movements are controlled, as well as arm muscles and sensory regulation of touch, for example. So there's an open question whether there is an additional transition zone where neurons have to make a kind of active decision to become dedicated brainstem neurons or cross over. And that is something that we look at in our research now. First, we wanted to know whether neurons do make this decision early in development. And for that, we injected a retrograde tracer into the peduncle, so in the brainstem, that labels all of the neurons that have an axon at this point in time, which is P0. So this is where we expect the axons to be here. This actively labels all of the neurons in the layer 5, which you can nicely appreciate here. And if our hypothesis is true that there is a subset of neurons that specifically projects only to the brainstem. If we inject the tracer on the cervical levels later on, we would expect that there is a subset of neuron that is labeled, and indeed that is the case. So we see that first here in the cross section, but also the quantification here. You can appreciate that in the lateral cortex specifically, a lot of the neurons are only labeled by the yellow tracer, not by the magenta tracer, showing that about 80% of the neurons actually become dedicated brainstem projecting corticospinal neurons. We also wanted to know if this decision is maintained into adulthood, and for this we did a so-called timestamp experiment where we switched on a TD tomato receptor, TD tomato reporter that we give in the cortex by injecting a retrograde tracer switch on molecule in the cervical spinal cord at P2. So this is 2 days after birth. And any axon that is here present in this area at this time point will get switched on and will express the reporter, and we can then count the axons on different levels throughout this throughout the neuroaxis, and we identified that there is actually a massive loss of neurons that projected onto the cervical spinal cord. So these neurons, like 90% of the neurons that were projecting to the cervical spinal cord at P2, now at P28, when we looked at this, are actually pruned back. So on top of brainstem neurons making the early decision to become brainstem neurons, cervical neurons now also decide to become brainstem neurons. This is especially interesting because in contrast to the cervical thoracic zone, as I mentioned in the beginning, we don't see this drastic reduction. So this seems to be a very brainstem specific thing. And to cut a long story short, we also have a handle on a molecular marker that projectively marks brainstem projecting neuron. As we can see, a double labeling only when with our tracer that we inject into the brainstem, but not when we inject it into cervical spinal cord. So to summarize this, additionally to the cervical thoracic transition zone that was previously established, we also now show that there is a brainstem cervical transition zone that is indeed established early in development, that the decision is maintained into adulthood, but that there is a massive amount of refinements, and that we have a molecular program that steers to this, and Kelch Like 14 will allow us in the future to look at the moleculars that steer the transcript. We can do transcriptomics to look at what exactly makes a brainstem neuron, a brainstem projecting neuron versus acervically projecting neurons, and it will also allow us to do some loss and gain of function and look at motor behavior as well as axon targeting. So stay tuned, and with that, I'm happy to take any questions.
Thank you, Julia, for the excellent talk. We'll just give people a minute to send in any questions they might have in the Q&A. You can also raise your hand and we'll invite you to come talk with Julia. We'll just give people a minute to take those out. All right, I think that we'll just take just a little bit more time, wait for people to type out their responses, but we can always allow people to ask questions throughout the duration of this talk. So if you end up having a question for any of the panelists, feel free to send those in. I do see that we have a question right now. This is from Kelvin Jones. If CSNC cells express Crim1, they will grow past the transition. Isn't this true for the CSNA?
So in this case, I am not entirely sure what they refer to with CSNA. I'm just going to assume that that's got maybe means CSNB, which could mean our brainstem projecting neurons. No, Carolyn, no.
Oh yeah, and Kelvin, if you'd like to clarify, feel free to, You can follow up on that Q&A or in the chat, and I'll just relay that. Or you can come on screen and clarify if you'd like.
Yeah, so far the experiments have been done. So this is the work that has been done mainly by my supervisor, Sahni. So I'll just briefly refer you to his upcoming paper, but generally overexpression of Crim1 does lead to to more axons growing into the thoracic zone. Of course, we don't know if these that do then finally grow over, what their molecular identity was initially, if this was a cervically projecting neuron, or if this could have been a brainstem projecting neuron beforehand. But that's definitely something that we want to look at in the future to find out which subtype of neurons can be used or can be manipulated to grow into a different target, because this will actually be really interesting also for spinal cord injury models, stroke models, ALS models, because the idea of this whole research is not only that we get, that we find out the molecular identity, but that we can maybe then in the future use those molecular identities to steer the regenerating neurons to their respective target that we want to get. Like, a lot of the currently applied regeneration models, all they're focusing on is getting more growth and more sprouting of the neurons. But if the neurons don't have a map in their hand, just letting them run doesn't really help anyone. So we want to get them to their targets, and this research will hopefully help with that.
Yeah, those are good. Those are great points. Kelvin was clarifying. Said they couldn't read the screen well. So yeah, they meant the cortical neurons that stop in the brainstem is what they were referring to.
Yeah, so I stand by my answer.
Excellent. Perfect. All right, we have another question that just popped up from Rebecca Blume. Sorry if I butcher names, by the way. Is this specific for motor neurons, or does it also apply to neurons mediating top-down regulation of sensory neurons in the dorsal horn?
Now I would love to have a clarification of what this means, if this is true. So this is probably the disadvantage of Zoom meetings. This would be easier to discuss in person. So I'm just going to assume that this specific targeting, it's not going to just be in the cortex. I mean, we also know from the Hox genes how certain neurons in the spinal cord get their own identification. And I am just going to go out on a limb here and say that for sensory, we can expect something similar. As it's a common pattern throughout development. So I'm just going to assume that bluntly, but I'm not aware of any research being done on this. So very interesting point though.
I agree. And that sounds reasonable to me. And Rebecca, feel free if you want to clarify that in the chat or to come on, feel free to do that. I see a question from Noah Golan. Hi, Noah. First of all, good to see you here. And she says, hey, Julia, lovely talk. I was wondering where the Crim1 thoracic/lumbar transition zone data were published.
It's not published yet. It's in the revision at the moment. So stay tuned. It's going to be out. Anytime.
All right. We look forward to that.
Yes. In the meantime, feel free to reach out to our supervisor, Sahni. I'm sure he's happy to talk about this research.
Excellent. All right, so I'm going to sit here. We